Hello and welcome to Chemspec Digital 2021. I have the pleasure to introduce the third session of the Innovative Startup Presentations, focusing now on the new synthesis routes and materials for sustainable business. Dr. Holger Banks of BCNP Consultants and European Chemistry Partnering will be moderating the session from here. Over to you, Dr. Banks.
Thank you very much for the kind introduction. Yes, it's our third hour, and we had already fantastic insights into different fields of entrepreneurship and innovations. For example, the presentation about the new European Circular Bioeconomy Fund with a strong focus on bioeconomy, but also the presentation about the Global Entrepreneurship Center by Friedrich Barth. He's building up a new facility to help not just bioeconomy, but also chemistry startups to grow and to reach the higher technology readiness levels. But also the insights by Maria Zano regarding the venture fund of the ECDF is very important to bridge the gap in financing and in production. And now we would like to give you in our final hour 4 presentations about really innovative startups from all over the world to inspire you, to bring up new ideas so that you can also contact these people and collaborate. You know, startups are very important regarding our future and the sustainability of our planet. And on the other hand, these young companies always need collaboration partners, finances, and so on to go further on. And so support our growth on Earth in a sustainable way while you are contacting startup and you're collaborating with startups. That would help us a lot, in my opinion. And if you have not enough after these 4 presentations, I'm quite sure we see us at the 6th ECP next year in February, which will be digital. I'm the initiator of this European Chemistry Partnering. It will be from the 16th to 17th February. And of course, there will be a meeting in presence at the end of May and the beginning of June. It will be the 35th Chemspec Europe in Frankfurt. We will see us again next year in the middle of the year in Frankfurt. But now enough words from my side, and we would like to start with our first startup, which is called 4Gene. And today with us is one of the founders, is Thilo Fischer. He's an expert in flavors, and 4Gene, it's all about activatable flavor and fragrance precursors. And Thilo is now giving us the story of 4Gene. So Thilo, over to you.
Thank you, Holger. Just let me share my screen. Okay, I'm Thilo Fischer from 4Gene, and I would like to present you our Glucosylation biotechnology platform. This is based on plant enzymes, glucosyl transferases, and these are perfect enzymes to do the job converting alcohols, amines, and thiols, for example, geranol, which is a citrus flavor, to their respective beta-D glucosides. But there's one small disadvantage. They are kind of specific. So if you want to convert a portfolio of compounds, you need an enzyme library. And it's also a wise option to do biotransformation, because this way you avoid using the chemical UDP glucose, but make the cells do the job. This was what we were busy about the last years, and currently we have some 50 products which are used for body care, industry, and pharma. I will show you now. And analytical standards are also now available via Karl Roth company. Most options are in body care and cosmetics, and what we use the glucosides here for is in a combination with a free flavor. So we use flavor glucosides, and they are set free by bacterial enzymes, and you combine to get a long-lasting effect. But in the end, it's also about a later effect. So if you do some exercise in the evening, there's also be a triggered response, triggered release of flavor late in the evening. So there are many options for these compounds. And what we set free, apart from the flavor, is a biological molecule. But later we became aware that's not only positive flavors which are interest for use, which can be used as glucosides, but there's also compounds with a warning character. And this is especially true for smoky flavor glucosides. And these have the ability to be pyrolyzed at a temperature of some 130 degrees. And we put them onto stickers, but there's also a pen solution for cables. Yeah? And if you apply such compounds in these forms at critical installations, you will get a overheating protection for your devices. And the warning is much earlier than with the smoke detector, because the smoke detector is dependent on the development of smoke at some few hundred degrees of Celsius. So you have additional pre-warning time, and you have additional safety saving your installations. So this is some kind of special applications, and we now turn to pharmacy, because in pharmacy glucosides always have been used, but now they are available by biotechnology. We already have 3 active pharmaceutical ingredients converted to their respective glucosides. One is paracetamol glycoside. The most important thing about these options, I think, is the bioavailability, which is improved in the form of glucosides. So they can be cleaved on the skin, in the mouth, in the intestine, and they serve as prodrugs. So you have a retard effect and a depot effect. But they are also there's also better water solubility and improved chemical stability. For these compounds where you can't form a glucoside, we have an emulsion-based solution. So we use cleavable flavor glucosides, which can deliver your active principle, for example, in the skin. But last not least, there are also legal options. If you convert your conventional active compound into a glucoside, you have the option to get a new patent for it and another 20 years of protection. So the time is short. I'm sorry for these technical problems. This is the founding team. There's much more information about it, and we would enjoy to share this information with you and discuss your options for your business. Thank you for your attention.
Thilo, thank you very much for this overview, and don't worry, complete understanding for technical problems. We survived it, and it was very interesting to see these insights into this multifunctional new class of compounds. So when you talked about 50 products, what does this mean? Is this already a final market product for the B2B or B2C market? Or do you mean you have produced from your glycosides 50 different types and you have some kilograms in your facilities?
Yeah, of course, you are right with your question. These are in quite different stages of development. Yeah, some we have the very first experimental proof that it's possible to make this glucoside. Yeah? And for some, we are able to make kilogram amounts for pilot customers. Yeah? So it depends, and so it's from the lab scale, scientific scale, up to 300 liter fermenter up now. But we are ready to scale up, and we would like to share this way with our customers. Yeah, maybe they have biotech facilities, and we can give them a license. So we have to decide with our customers how to go this way.
Okay, and you are active in quite different markets. What about regulations regarding your chemicals?
Yeah, we have filed the application for menthol glucoside for food at EFSA. We haven't got a decision yet. This, I think there was some delay due to Corona. For cosmetics, it's possible. We rely on referring to the free flavor. This is common. For example, if you use flavors like aldehydes, the pro fragrances are Schiffsche Base, yeah, Schiffsche base, yeah. And it's common to refer to the flavor which is set free, yeah. And they do the same for the glucoside. Yeah? And the technical use, of course, is free. Yeah? All flavors we use, also the smoky flavors, are already in use for roasted beef, for example, or roasted fish. So they are applied in food, yeah? And we just evaporate them in minor amounts. Yeah?
Okay.
So there's no problem with this.
Thilo, thank you very much for these insights in your company, and all the best for you regarding future collaborations with the big ones.
Yeah, thank you for making this possible. Thank you.
Thank you. So our next presenter is Wolfgang Eger, and he is presenting the company Iosynt Labs and biocatalysis and fermentation from India. And we are waiting for him, but now he's coming. And here he is and his presentation. Wolfgang, over to you.
Hello, everyone, and welcome to my presentation. I'm going to introduce you to our company called Iosynt. I'm one of the co-founders at Iosynt, and I'm responsible for marketing and international sales. Iosynt is, as Holger just mentioned, based in India. We are a Bangalore company, and we are now I have the same problem as my. Just a moment. I also have the problem with my screen. Just a moment. Okay, we have to do it this way. We are active in the field of biocatalysis and fermentation. Iosynt applies biology to produce sustainable chemistry. And if we say apply, then basically we mean we are focusing on large-scale industrial production of our technologies. To make that distinction a bit clearer, we are not academic, we are not deep tech research organization. Our R&D scientists focus on developing robust, scalable processes which are ready for a plant implementation today at multi-ton scale. Our core technologies are biocatalysis and fermentation, and we combine that with traditional organic chemistry wherever it makes sense or where a process needs an additional chemistry step to come to a useful end product. The combination of these core technologies help us to achieve great cost effectiveness and a better sustainability. So it dings on both of these key parameters. This allows us to produce 4 different product categories. One is enzymes and wholesales. The other one is pure fermentation products, which are not enzymes. Then there's bioprocess intermediates and, of course, pure chemical intermediates. Each of these projects or products we can take from a very early project inception to proof of concept, optimization, upscaling, and large-scale manufacturing. So we cover the full product development cycle that we would need in order to have an end-to-end process. Our company offers products and services in this field. So in terms of products, our main products are enzymes and intermediates. In enzymes, we offer mainly biocatalysis enzymes to make these chemical transformations. This includes screening kits, but also bulk enzymes and enzymes with specific application protocols for fast implementation of specific technologies for specific intermediates or APIs. The intermediates are the combination of our technologies, specific intermediates to produce, an API in a sustainable way. Our services focus on the 3 technology platforms that I mentioned before, and it basically offers pretty much any of these services that you need in these technology platforms. We offer the services both in a CRO model or a CDMO model, whereas we prefer the CDMO model where we can also produce the end product at the end of the journey. I'd like to talk you through 2 project examples. So these are common examples that we have seen with existing customers. The first example is where a customer has a fermentation strain but there's nothing else developed on top of that. So sometimes customers develop these fermentation strains themselves, or they get it developed, and then they still need a process development to ferment the strain, but also to isolate and upscale and manufacturing that whole process. So these are one of the nice projects that we'd like to have. We take the strain and then develop this optimized fermentation process on top of that, including a full downstream process development and stage-wise upscaling through pilot all the way to multi-ton fermentation scale. This we can do for enzymes, any enzyme class that you can think of, but also for SynBio-derived fermentation products, which could be chemicals, flavors, fragrances, pharma substances, proteins for food or feed. The second example is where a customer has a theoretical reaction in mind. So very often they come to us and say, here's a reaction from A to B. I know that this could be very elegantly and nicely done with an enzyme, but I have no idea which enzyme to use, and I don't have a process or any of that to take that to production scale. So in this case, we look for the enzyme, do a screening, create a proof of concept, and then also develop a process on how to utilize the enzyme and then how to isolate the end product that the customer wants. This kind of customer projects very nicely combines our different strengths in the technologies, anything from enzyme immobilization and chemistry. Typically it's very difficult to predict but what is needed. But at the end of the day, very often a lot of those technologies are needed. Two cases very quickly to wrap up here. These are intermediates. Both of these cases are intermediates that we are currently upscaling to a ton scale. One is where we introduced this chiral center here with a ketoreductase. It's an intermediate for a pharma API, and it's done with a purified enzyme at very high conversion, very high yields. It's a very nice process that works also very fast. The second molecule is where we introduced a carboxyl group here, and this is a wholesale process, so it's not a purified enzyme, and it's a bit more cost effective in this case, but it also gives very nice yields and efficiency rates. That was all that I wanted to share with you today. Thank you for the attention, and if there are any interest, please reach out to me. I'm happy to have a deeper discussion with any of you.
Wolfgang, thank you very much for this interdisciplinary combination of fermentation and organic chemistry. And you mentioned you are producing in ton scale already. Do you have then already partners for these special molecules, or do you that by your own?
There's a very good question. There's two ways to that. One is sometimes the second case study that I presented was a customer-driven project. So the customer has a plant that we can utilize to implement that technology. So it's a purely customized-driven project. We develop the process, and then the customer produces that finished product. But one of our investors is also linked to a big Indian pharmaceutical company. So we have our own access to facilities as well.
Okay. Wolfgang, thank you very much for this overview and all the best. And I'm sure we see us next week, maybe at ECP or Chemspec Europe.
Thank you for the opportunity.
Wolfgang, thank you. So the next in our row is Christoph Vetter. He's presenting WESH Technologies from Germany, dedusting solution filter systems. And here he is already.
Exactly.
Christoph, thank you very much. Over to you.
Thank you for having us. Good afternoon. And first of all, my product is much easier to understand, but not less needed. And I will also try to start my presentation because I'm showing two video files. Let's see if I'm more successful in my procedures. Yes, this looks good. Okay. What we are doing is providing dedusting solutions for the pharmaceutical industry because our founding idea was to provide a fitting vacuum cleaner for a pharmaceutical tablet press. And during our company founding, we've found that a lot of companies in pharmaceutical, but also in special chemical industry, need some not off-the-shelf solutions for their dust problems. So whenever you're producing something for OSD or some special powders, we are the go-to guys if you have to handle with some dust which are either inflammable or toxic. So what we're basically doing is we are selling the most, the most expensive and also the most functioning vacuum cleaner on the market because we are focused on these three topics. The GMP area, I don't think it's too much interesting for the chemical industry. That's why I'm skipping it in this time. But we are producing systems. I can just show you like one in the back, which can be placed in a clean room, which have the proper documentation of the materials and so on, and which are also designed to prevent cross-contamination. So if you have a GMP production, you have to contact us. In all other cases, we would like you to. And we are concentrating on highly toxic dusts and on inflammable dusts. So the problem with HPAPI, so the high potential pharmaceutical ingredients, is that they do what they do. They're working in the human body. But when you're producing them, you don't want them to work on your employees as well. And the main principle is that you have to use the stop principle for protecting your employees. You can't substitute your material because you want to produce it. So the next step would be to take technical measures before organizational measures, and to simply put your employee into PPE, into personal protective equipment, should be your last step. But it's not as easy as it sounds, because as you can imagine, you can easily put glass walls to a tablet press and then it's isolated. But when it comes to the suction of the dust, you need to empty the dust discharge container, and you also need to change the filter cells, and you still need to have the technical measures in place to protect your personnel. And just to give you an overview, for the last years, respirable dusts were like the most difficult to handle in the chemical industry, and maximum value was 1 milligram per cubic meter air, which is in the nice containment pyramid which I brought you, the second lowest level. So pretty easy to handle. But within the last years, for most of the materials, the requirements increased. So we are having now a project with lithium dust, which is already by 200 microgram per cubic meter air, so 1/5 of the former highest level. And if we go to the pharmaceutical ingredients with HPAPI, for example, cancer medication, then you even go to an area which is at 2 microgram per cubic meter or something like that. And the problem is all of your equipment needs to be better than your OEL. So if I want to handle 2 microgram per cubic meter air dusts, I need to have a system which at most exposes 1 microgram per cubic meter. And just to give you an example of what this means, this is the Olympic pool in Munich. And the Olympic pool in Munich is quite big. But the highest level of containment which we are providing is like a pinch of salt inside of 12.5 Olympic swimming pools. That's the level where we are operating. So whenever you are seeing dust at one of our units, it's already too late and you're already poisoning your employees. And as if toxic dusts weren't hard enough to handle, we deal with another problem. because most of the dusts are also inflammable. And what happens if you are handling inflammable dusts the wrong way is shown here. They simply explode. So we are we have the problem that we have to provide sensors, we have to provide the equipment, all rated for an ATEX environment. Sometimes the whole production facility is ATEX rated. So we need exposure protection inside of our filter system and also outside. And it's pretty rare to find filter systems, especially for higher volume flows, which can provide something like that. Some vacuum cleaners have that, but filter systems are pretty rare. Yeah, and with those two major, or three if you count the GMP environment also in, with these two tasks, we started our company in 2018. And ever since, we experienced that it's quite a needed product, even though it's not as sexy as something you can produce. This is a picture of one of our first fairs, showing our laboratory dedusting system, which stores the dust simply in a filter cell, and also our compact dedusting system, the system which my co-founder is showing, contains of a cyclone and a HEPA filtration unit, and therefore you can also reuse the materials which are collected. Yeah, thank you very much. I hope it's interesting for you as well. I don't know if anybody is using the production as well, is in charge for the production as well, but thanks for your time.
Christoph, thank you very much for this completely different view on valuable chemicals. So I like this very much because it was a completely different perspective you gave us. And coming once more back to the collected material which you have in your filters, do you give then the filters back to the customers or do you recycle them by your own, for example, regarding the lithium material?
Normally, our customers handle the disposal of the filter cells themselves, so they can choose what to do with it. But if you have toxic materials, you don't want to open the containment at any place. So if you have the valuable but non-toxic materials, we always recommend to have a unit with a cyclone in place, because this way the valuable materials simply drop out of the cyclone and can be collected in a stainless steel drum. And then you can simply re-add it to your production process. It has only touched stainless steel to this point. And if you use one system for one chemical material, it's still pure when you recollect it from our cycle. Okay.
Christoph, thank you very much for this additional explanation and for giving us this talk, and all the best for you and your company.
You're welcome. Thank you.
Thank you. So, to the audience, we are nearly at the end of our third hour and our startup session at this year's Chemspec Digital. We have one final presentation in a new mode because it will be a video, but I would like to say thank you for being with us this afternoon. I found it really very interesting, and a very, very big thank you to our two keynote speakers, Maria Asano from the European Circular Bioeconomy Fund, and also Friedrich Barth from the Global Entrepreneurship Center, and to all you presenters from the young companies, young and growing companies, and you inspired us and the audience a lot. Thank you very much. We will see us next year in February at Digital 6th ECP, and we will see us end of May, 31st May and 1st of June at Chemspec Europe number 35. And now I would like to introduce our final speaker, and that's also the end for me because you will then see the video, and it's Juan Maria Gonzalez Caballo, and he will present Rochade Research Services from Scotland. Catalysis and material science are some buzzwords for you. And now, please show us the video.
Good afternoon. I'm Juan Maria Gonzalez Carballo. I'm a process scientist at Rochade Research Services. On this short presentation, I'm going to introduce our company to you, talk about our activities, our interests, our capability, and finally show a couple of examples showing how we have helped two of our customers to accelerate their innovation. Rocket is a contract research organization with the expertise, experience, and capability to carry out world-class work in the fields of homogeneous and heterogeneous catalysis and material science. Our customers choose us because we are the perfect partner to accelerate the development of new technologies and products or improve existing ones. We have a flexible approach and a track record of applying our resources to straddle different sectors, including chemicals, materials, energy, circular economy, and bioeconomy. We have a global customer and partnership base, with clients in North America, Europe, and China. Rocket is the Scots Gaelic word for bridge. That reflects the bridge we build between invention and commercialization. We pride ourselves of bringing the right solutions at the right time. Our main activities focus on de-risking, scaling up, and commercialization of processes. We have a team of 9 scientists with extensive industrial experience in petrochemistry and allied industries. We have first-hand experience in the development of world-scale chemical processes. We comply with industrial standards and procedures and also work closely with those engineers and others involved in commercialization processes. Our work relies in 4 strategic pillars. De-risking process innovation, catalyst testing. We generate the critical process data to de-risk decisions to progress to piloting of new technologies. We provide rapid parallel screening of numerous catalysts and or varying reaction conditions, as well as long-term trials. We have the ability to synthesize a broad range of materials, including organic molecules, organometallics, and nanomaterials. We have state-of-the-art computational methodologies and synthetic skill base. These allow us to develop and bring to fruition strategies for the rational design of functional materials. We believe that to understand your process, you need to understand your catalysis. We carry out process optimization through detailed characterization of functional materials, including under operating conditions. Finally, we have also the ability to analyze, to unravel the structure and composition of feedstock and complex mixtures. That includes product excellence of refinery processes and also from biomass transformations. On these 4 examples, I'm going to briefly describe one work we've recently done with a biotech company. It's about process optimization of biocatalytic transformations. Also, Biotrans is a biotech company who choose us to optimize some of the parameters of the process. Specifically, in this case, they ask us to refine the concentrations of the enzymes used to produce one of their natural products. The optimization carried out by Rocket not only improved the efficiency of the technology developed by our partner, but also helped them to progress on the development and scaling up of their process. This second case study is about technology validation and kinetics for scaling up. In this case, we partnered with an specialty manufacturer of textiles and a startup company which were interested in finding a bio-based chemical to replace the one that they were currently using on their process. Partnering with Rocket allowed our partners to validate the concept that they were working in to accelerate the development of their process and also provide the kinetics for large-scale production of the bio-based chemical. Just a brief summary of the slides I've just presented. We have extensive process and product experience to solve complex challenges and the risk implementation of novel processes and products. We believe that Rocket is the perfect business partner to deliver the full potential of innovation. Please find out more in our website rockets.com or email us at the address that is shown on the slide. We are always happy to discuss ideas or projects or concepts and try to find out how we can support or help you to accelerate the innovation. Thank you.